Hello guys and welcome to MedExpert. Today we're going to talk about the structure of the amino acids and how easily we're going to be able to draw the structure of the different amino acids. So in order to understand the structure of the amino acids, first of all we have to understand a very important aspect of the amino acids, and this is the two categories of the amino acids that basically you're not going to see it in most of the books or most of the videos that you watch. They don't even indicate on it, and most of the people, they don't even know those structures. They are amino acids. They just know them as neurotransmitters or some chemicals between the synaptic cleft, or something as a what I call using as a supplement, like for workout or something like that. But basically those are the amino acids as well. So the two big categories, they are non-proteinogenic amino acids and proteinogenic amino acids. Okay, non-proteinogenic amino acids. Basically, the non-proteinogenic amino acids, those are the amino acids that they're not going to go to the biosynthesis of the proteins. One of the examples of them is beta-alanine. Beta-alanine, basically, it's used in the pre-workout powders, and usually the athletes, they use them because it's going to give you a little bit more pump and it's going to give a little bit more energy during the workout. The other one is 4-amino benzoic acid that you see it in the picture, and Gama Amino Butyric Acid, which is GABA. GABA is one of the most important one, and GABA is actually a neurotransmitter and a receptor as well in the synaptic cleft. That what it does is going to regulate the signals between the nerve cells. That means when the nerve cells or the neurons, they are shooting the signals from one neuron to another neuron, that's going to be very, very fast. And if with that speed goes from one neuron to another neuron, it's going to damage the other neuron and it's going to kill the brain cells or the nerve cells. And that's why it's so important to get regulated. And the GABA, which is Gama Amino Butyric Acid, is the one that is going to help to regulate those signals and slow down the speed of the signal and send it to other neuron in order to inhibit that. Then I'm going to come back to the structures and I'm going to teach you how to draw these structures so easily without even like forgetting about them. Okay guys, I know that most of you guys, when you saw the title of my video, which is saying how to draw the structure of the amino acids easily, all you were looking for is this 20 amino acids, which all of them, they are proteinogenic. So these proteinogenic amino acids, those are the 20 main basic amino acids that we need them in order to biosynthesize the protein. So in order to learn the structure of the amino acids, first of all, we have to understand something very important, which is the chirality. So the chirality, it's one of the main important part of the story in learning the structure of the amino acids. What does it mean by the chirality? Let's just like make everything simple. The chirality means 4 different groups are connected to the carbon atom, and in this case, the alpha carbon. The only achiral is glycine. Okay, we're going to explain it exactly what's going on right over here. Probably some of you guys are getting confused with these kind of stuff, achiral or chiral, or probably with this alpha carbon and everything like that. So in order to understand, we're going to pause the presentation here and we're going to go to the whiteboard, the main part that we're going to start actually draw the structure of the amino acids. Okay, great. In amino acids, basically its name says half of the structure. What do I mean by this? Look, amino acid, that means like there's amine and there's one acid. So this is the half of the structures, because we know that there are 4 groups in the amino acids and we already know the 2 of them, and the 2 of them is going to be amine and acid. So how the acid is going to look like in the organic chemistry? What is the acidic group? Yes, that's right, carboxyl group. And how you draw the carboxyl, how you actually just write the formula of the carboxyl is going to be COOH. This is a carboxyl group. Or sometimes you're going to see it like COO minus. It doesn't really matter. It says one thing, and it's a carboxyl group. That's all. And when we come to the next one, which is like a amine group, it's going to be NH2, or sometimes you're going to see that like NH3 plus. it doesn't really matter. You can go with the both extra structures, and the reason is actually they put it sometimes like a COO minus or CCO or NH3 positive is because of these Zoeterian, we call it the structure that we're going to talk about later on. So in order to understand it better and try to draw the structure so easily, first we're going to put one carbon in the middle because we know that there's a carbon in the middle. Then we're going to connect the carboxyl group to it. So there's a double bond right here and the OH. At the bottom, we're going to go with the amine group and connecting the 2 hydrogen to it. All right, half of the structure is already done. This is a general amino acid structure. And the third one, it's so easy. It's just a hydrogen. That's all. You just need to add the hydrogen to that group. And the fourth one is an R group. And what is this R group? This R group could be anything. It could be like a methyl group. It could be a ethyl group. Or it could be like a sulfur. And we're going to see that that's the area, this R group actually makes a difference between the amino acids. That means the amino acids, the difference of the amino acids comes from this R group because everything, rest of the things is the same thing. Okay, here we're going to talk about the carbon. Why this carbon called alpha carbon? This carbon called alpha carbon because it's connected to the group that has the higher priority. Okay, I know guys, some of you guys don't remember those higher priorities based upon the atomic number, but don't worry about it. I'm going to tell you exactly how it works. I know that you guys are thinking about the higher priorities and based upon the atomic number. Yes, this is very important to know because that's how we're going to be able to understand the mechanism of these amino acids and how they are being built. Alpha carbon is the first carbon that is connected to the group, to the functional group with the higher priority. So look at here, which one has a higher priority? Carboxyl group, hydrogen, nitrogen here from the amine group, or the R group? In this case, the carbon, alpha carbon, is connected to the nitrogen, and the nitrogen has a higher priority. So the first carbon that is connected to the higher priority group is called alpha carbon. And if you want to remember how these things works, you have to know first it comes sulfur, then we have the oxygen, then we have the nitrogen, and then we have the carbon, and at the end we're going to have the hydrogen. That's how it actually works. Okay, but there's one problem actually. Some of the students, they're going to confuse this carbon right here with alpha carbon. And they say, why don't you choose this one? Because this carbon is part of the functional group carboxyl. So it cannot be alpha carbon. This is the alpha carbon because it's connected to the N, and N nitrogen has a higher priority than the C, which is carbon here. Okay, so this molecule is a chiral molecule. Why is it a chiral molecule? Because 4 different groups that are attached to it. If you look at it here, we're going to have a carboxyl group, we have hydrogen, and we have here a amine group and also the R group. So this is a chiral molecule. Now that we know these, let's go and get to the structure of the first amino acids. Before we go through the structure of the amino acids, we have to know something very important about the structure of the amino acid. Because amino acids, the first and second or probably third amino acids is going to be easy to draw if you are choosing any type of a structure, for example, the Lewis structure or the line structure. But when we get to the higher actually priorities and when we get to the bigger amino acids, it's going to be harder to actually draw the amino acids in the Lewis structure. The Lewis structure, if you remember, for example, we're going to start with the structure of the first amino acids of the proteinogenic amino acids, and we're going to start with the aliphatic nonpolar amino acid called glycine. So the glycine, we're going to put the alpha carbon in the middle. We're going to put the carboxyl group on the top. All right. And then we're going to put the amine at the bottom. The hydrogen here. And the glycine has another hydrogen connected to it. So first of all, I'm going to tell you something about the glycine. Glycine is the only amino acid that is achiral. That means two identical groups right here that are connected to it. That means if you pay attention, these two, they are the same, two hydrogen. That's why it's going to make it achiral. This molecule is not chiral, and this is the only amino acids on the planet which is achiral. So if you look at this structure, it's so busy and it's just like too much hydrogen here, showing all the carbons and everything like that. So it's not going to be a very scientific one. It's good, it works, you know. If you remember, when we get to the harder one, it's going to get very hard to remember everything. So the best one and the most scientific structure of the amino acids is line structure. Basically, line structure is going to be the easiest one and the most scientific one that you're going to see it everywhere when you're trying to draw the structure of the amino acids. And this is very, very important and very nice to learn. So in order to draw the structure of the first amino acid, which is glycine, in a line structure, we just need first to put the double bond of the carboxyl group, connecting the OH, then connecting the NH2, and basically our structure is done. Sometimes, for example, the people, they just want to make it a little bit look better, or just trying to show that there were hydrogen here, they just add two hydrogen like that to it. But this is what we call it now, glycine. You see, it was so easy. And all the amino acids that we're going to draw from now on, they're going to have this structure of the this common structure right here. That means the area that we got connecting like a carboxyl group and NH2. If we want to interpret the line structure for you guys, is very easy. Look at that. I'm going to draw the structure of the glycine again. Double bond, OH group, coming down, putting an NH2. What exactly mean when we are doing something like that? What are these actually junction means? These junctions means we have one carbon here, we have another carbon here, we are just hiding the carbon and hydrogen. That's all. That's all we are doing. We don't need to draw those because we are basically knowing that what's going on over here. We know that the carbon is here. We know that the hydrogen are here. We just don't draw them because we don't want our structure to get complicated and very crowded. So you see that how easily we're going to be able to draw the first amino acid, which is glycine. Okay, let's talk a little bit, because we said that in this lecture, we're going to talk a little bit about the clinical correlations of the amino acids as well when we are drawing the structure. So we're going to talk about the glycine a little bit. Glycine is going to be the smallest amino acid that exists, is non-polar based upon the chemical properties that it has. And it's one of the smallest amino acids that I told you. And this being small and being kind of like a amphoteric, that means like it can be basic, acidic, it depends, you know, it can work and it can act in both sides. So glycine also has an amphoteric property, so it's going to fit almost in everywhere in the structure of the proteins, and that's why it's going to be very essential in the structure of the collagen, which is so important for our tissues and skins. Also, it's very, very important as an inhibitory neurotransmitter.